In this video, we're going to continue to look at how the organs are involved with metabolism. And so we're going to talk about hexokinase and glucokinase now. And if you remember, hexokinase is the first enzyme in glycolysis that takes glucose to make G6P. Well, liver has a different enzyme that does the same thing called glucokinase. So, when your blood reaches high levels of glucose, that is 6 millimolar or higher, your liver will start taking this glucose out of your bloodstream and convert it to glucose 6 phosphate. That's what glucokinase does. So, if we look at hexokinase and glucokinase, they have Michaelis-Menten kinetics. Hexokinase has a much lower Km of 0.1, compared to glucokinase, which means that hexokinase is active at much lower ranges or much lower levels of glucose. However, glucokinase can increase rapidly when blood glucose gets over your normal ranges. And what's nice is that glucokinase is not inhibited by G6P. Hexokinase can be stopped if you have a lot of G6P around. Glucokinase will keep working. And the reason why these enzymes are designed this way is that if your blood has low glucose, you don't want your liver to take glucose from your blood for storage. You want your cells to take this glucose. So since glucokinase has a Km of 5 millimolar, it will not compete for glucose at low glucose blood levels. However, if your blood levels of glucose are high, that means your tissues are well fed. So glucokinase will now start taking glucose from the blood and will use it as storage. So glucokinase is controlled. It's controlled by a protein called glucokinase regulatory protein, so a name that makes sense, which can inhibit glucokinase if there's F6P around. However, fructose 1-phosphate will overcome that inhibition from F6P in glucokinase regulatory protein. And the idea there is that possibly when you eat fructose, this is a sign that the liver should uptake glucose. They should start storing that glucose. Now, continuing on with glucose 6 phosphate, it has a lot of different pathways it can take in the liver depending on what is going on with your body, what is the glucose demand. So first off, we can take that glucose 6 phosphate and just convert it to glucose and send it off into the bloodstream. This will happen when glucose levels in your blood drop. When they're less than 5 millimolar, your liver will start releasing glucose into the bloodstream. Also, when your blood glucose levels are low, that G6P will be turned into glycogen for storage. Now, G6P can also be converted to acetyl-CoA via glycolysis, and this acetyl-CoA can be used to make stuff. It can be used to make ATP, as we talked about, or fatty acids, phospholipids, cholesterol. So that's another fate of G6P. Or, one last fate, G6P can go through the pentose phosphate pathway, generate NADPH for biosynthesis. And we have talked about the majority of these pathways up to this point. So none of that should be a real surprise, just more of a refresher for you. Now, continuing on with the liver, since it's so important, fatty acids in the liver will be broken down to acetyl-CoA in ketone bodies if the demand for fuel is very high in your body. However, your liver can't use these ketone bodies as a fuel because it lacks a specific enzyme to do that. But what we can use, what the liver will use, is those fatty acids will be broken down to acetyl-CoA through beta oxidation. So fatty acids are the major acetyl-CoA producer in your liver. Now, if demand for fuel is low, these fatty acids will be secreted as VLDL, and your adipose tissue will take these up from the bloodstream and use them for storage. And the fatty acids that you make inside your liver won't be broken down into acetyl-CoA in your liver, or you'll just have a fuel cycle. Now your liver can also work with amino acids to break them down into various intermediates, or you can completely oxidize them to carbon dioxide and water, or you can make them into glucose or ketone bodies depending on what signals and what state your body is in. And talking about energy still, so if you're fasting, your muscles will be broken down to alanine and glutamine to make glucose, because as we talked about in the last video, your brain needs a constant source of glucose. So you can see all of your organs are connected into this giant web using all the pathways we have talked about so far in this class. Now talking about the last organ here is your kidney. That's going to take urea that we talked about, the urea cycle and other waste products, and filter those out of your blood. It will also make sure not to waste any important metabolites like glucose. It will help to maintain your pH of your blood by recreating your blood buffer, and your blood buffer is made out of bicarbonate. Any excess acid will be shipped out with excess with conjugate bases. The ammonia will be filtered out, as we talked about in the urea cycle. And starvation, your kidney can actually produce glucose as well. As we can see in this picture, glutamine will go to alpha ketoglutarate, which will go to glucose. So your muscle is breaking down proteins, which can be turned into glucose through alpha ketoglutarate in your kidney, and then it can be sent to your brain and other places. So your kidney is just not a factory for getting waste. It can also produce some energy for you as well. And that is that for this video. I will see you in the next one.